RAPD stands for Random Amplified Polymorphic DNA. It is a molecular biology technique used to analyze genetic variations in organisms. RAPD is a PCR-based method that amplifies random segments of DNA using short, single-stranded DNA primers with arbitrary sequences. By comparing the amplified DNA patterns between different individuals or populations, researchers can gain insights into genetic diversity, genetic relatedness, and evolutionary relationships. Here's a brief overview of the RAPD technique. DNA extraction. The first step in RAPD is to extract DNA from the organism of interest. Various methods can be used to isolate DNA, depending on the organism and sample type. PCR amplification. The RAPD technique utilizes PCR, Polymerase Chain reaction, to amplify specific DNA regions. In RAPD, short random sequence primers are used. These primers can anneal to multiple sites throughout the genome, resulting in the amplification of random DNA fragments. Gel electrophoresis. After PCR amplification, the products are separated using gel electrophoresis. The amplified DNA fragments are loaded onto an agarose gel, and an electric field is applied to move the DNA fragments through the gel. Smaller DNA fragments migrate faster than larger ones, resulting in a pattern of bands on the gel. Visualization and analysis. The DNA bands on the gel can be visualized using various techniques such as staining with a DNA binding dye or radioisotope labeling. The resulting banding pattern represents the genetic fingerprint of the organism. These patterns can be compared between different individuals or populations to identify genetic similarities or differences. RAPD is commonly used in various fields of research, including population genetics, evolutionary biology, plant and animal breeding, and forensics. It provides a rapid and cost-effective method for assessing genetic diversity and studying genetic relationships without prior knowledge of the specific DNA sequences involved. However, RAPD has some limitations, such as the possibility of generating non-reproducible results and the difficulty in interpreting the exact genetic changes underlying the observed